talk today about really 20 years of oyster research that we've done here in the Gulf. Anybody that doesn't know me, I'm Dr. Smee. I'm the chair of university programs here at the Sea Lab, and I'm a professor of marine and environmental sciences at South. So, my research, I'm a sensory ecologist at heart, okay? And so, what I'm really interested in is to look at how animals acquire critical information that they need to forage, to avoid enemies, to find mates, and the things that are, really necessary for their survival. I'm interested in the types of signals that they use to do that and how they integrate cues. Like if they see something and smell something, is that different than if they just see it or smell it? And how those things work together. And I'm also very interested in how the environment modifies organisms' ability to gather this information. For example, if the environment is really favorable for predators, it may make them really successful foragers. Uh, or conversely, it may make them unsuccessful foragers, which can then affect the distribution of their prey in a habitat, right? Based on how effective they can forage and how effective those prey can avoid being eaten. And this ultimately affects the structure and function of communities, as well as how they respond to disturbances, which I'll talk about today. So I do a lot of work on oyster reefs. I'm not going to spend a whole lot of time telling you guys that oysters are like really important and really valuable, and we care about them, and also that they're probably the most endangered habitat, definitely marine habitat in the world. Okay, so a bunch of them are destroyed, and so there's huge interest in rebuilding them and restoring them to recover these lost ecosystem services. And so I'm going to talk about two things today with the goal of understanding oyster reef ecology better to help with conservation and restoration efforts. So we're going to look at community structure and stability, including the roles of predators and disturbances and how biodiversity affects those things, the responses to those things. Then we're going to shift gears and talk a little bit about the chemical ecology of oyster reefs and how predators affect oyster morphology, the chemical cues that are involved in oyster detection of predators, and then finally how manipulating oyster morphology can help improve restoration. So I showed this picture of these classic keystone predators, right? So the sea stars are Payne's classic work in the 60s. Sea stars prey on mussels, which are the competitive dominant, and that creates higher biodiversity in rocky intertidal systems. And of course, otters eat urchins, which eat kelp, and then they maintain the kelp forest. And so in marine systems, I think we've appreciated the large role that predators can play for a really long time. And I like to reference this paper that Ken Heck and John Valentine wrote back in 2007 that summarized a lot of this, that in these shallow coastal benthic ecosystems, predation is usually really important and plays a big role. And when we look at things like wolf reintroductions into Yellowstone, we see that this top-down control from predators can have really big, large-scale effects on really big spatial scales in aquatic and terrestrial systems. And for you younger faculty and for you graduate students, when I tell you predators are important, that's probably not surprising to you. But through the 90s and early 2000s, there was a huge debate about what role predators actually played, right? And if those effects were really limited to aquatic systems with really linear food chains. In other words, are predators or these top-down forces important in systems that are really diverse and that have lots of species interactions, like oyster reefs? And so we thought this was a good system to test some of these effects, and we started this all the way back in 2007. So an oyster reef food web, anyway, can be quite complex and have a number of kinds of interactions. For example, on the bottom trophic level, we have the oysters themselves that of course build the habitat. We have porcelain crabs, which are suspension feeders, and they maybe at some level even compete with the oysters for food, but they take things out of the water. And we have grass shrimp, which eat algae that grows on the oysters and eat detritus and things around the reef. These are preyed on by things like snapping shrimp and mud crabs and maybe small blue crabs, which are in turn preyed upon by larger adult crabs and a variety of fish. So you can see already there's quite a few interactions here. But it's even more complicated than that. We have birds that come onto the reef and that prey on all of these things. We have, gobies and blennies and small fish that live in the reef. Um, there's intra-gill predation because a lot of these crabs are cannibalistic and they eat each other, and they can change trophic levels based on their size and their age. Okay, and there's omnivory here. The things don't necessarily just eat the trophic level below them. They may feed 2 or more trophic levels down, and some of them even feed on the oysters themselves, which are the habitat builder. So this is a really, really complex habitat with a lot of interactions. And so it seemed like a good place to test this idea: are predators important in diverse habitat? Well, how do you do that? Well, this is ecology, right? So we built a cage. Um, and we did it at Goose Island State Park in Rockport, Texas. We did a lot of this work here because the park was really good to us. We could put things there. The reefs typically aren't harvested there, so our equipment was safe. And it was just a good place to work. And we started this again all the way back in 2007. So we built these wooden frames that we call bio boxes. And then we cover these frames in mesh to exclude predators, or we leave them open for controls. And we've done a variety of different designs, and we've done all the cage controls with tops open and sides open and all those different kinds of things. And we put these out in the field in mid-April before the oysters do their first big spawn, and leave them out there until about the middle of June and let the community develop. We then use this throw trap to collect all the critters that are in the bio box and recover them. And I made a video that sort of summarizes or shows what we're doing. So here you can see in there is one of our bio boxes. We're going to pick up one of them here and put it in this kiddie pool. You got to work fast to be in my lab, by the way. So once we've got that, we use this bar seine, sweep it through the throw trap to catch any fish or crabs that might have drifted out of the cage. Here's a toadfish and we'll just keep doing that seining until we don't catch anything. And then we pick through all the shells and collect all the little crabs and critters and other things that we find there's a go-to. So Keith Johnson was one of my first PhD students. This was a huge part of his dissertation. And essentially what we found was that whenever we excluded predators, right, we got a significant change in oyster eats. Essentially, a lot of these intermediate predators, these mud crabs, became really abundant, and they were able to forage inside of our bio boxes without being controlled by fish, which ultimately led to a decrease in biodiversity because they just ate everything. We did not find a trophic cascade all the way to oysters because it turns out oysters in Texas, their spawning is really controlled more by salinity and other abiotic factors. And so some years they spawn a lot and some years they don't. But the rest of the system responds very clearly to predators. Well, then we wanted to know, well, okay, well, predators are important, but is it fish or is it crabs, right? And how could we design an experiment to test that? So to test the effects of fish predation, we really needed to find areas where fish predators were abundant and successful and compare those to areas where they weren't. And luckily, this is when Jessica joined the lab. And so Jessica wanted to look at turbidity. Most of these sport fish are visual predators. And so if the water's turbid, they can't see very well. They don't forage as well versus if it's clear where they can. And even at modest levels of turbidity, there's enough light scatter that it can interfere with their foraging. So the first thing Jess did was she went through Texas Parks and Wildlife's fisheries independent data set. And she just asked a question like, are fish more important when turbidity is high or when it's low? And the same for crabs. And what she essentially found is fish are significantly more abundant when turbidity is low, which makes sense because they're probably wanting to be in those areas because that's where they can forage well. And then crabs are a lot more abundant when turbidity is high, right? They're chemosensory foragers, and so they don't really care about turbidity very much, but also they have a refuge in that from fish predators, right? So it explains that. We also tested this on oyster reefs using bio boxes, and again, it showed this pattern where crabs were more abundant where it's turbid. But what Jessica also did that was really clever was she did this tethering experiment where we tethered crabs by supergluing them to fishing line and tying it to these landscape staples and put them in the field and looking at predation rates. And so we were able to look at predation rates in high and low turbidity, but also with crabs, you can tell what ate them in a lot of the circumstances because when fish eat the crabs, they tend to grab the whole crab and take it. When other crabs eat the crab, they often leave the carapace behind. So we can count the number of carapaces and know what ate those particular crabs that were tethered. And not surprisingly, Jess found many, many more crabs are eaten by fish in low turbidity. Okay? So turbidity is a really important part of the oyster reef predator story because it controls fish, which seem to be responsible for a lot of the predation that affects oyster reef structure. Now, this is Joey Riceley, and when Joey joined the lab, we were talking about next steps. And so we took the predator exclusion experiments that Keith had done, we combined it with the turbidity work that Jessica had done. I teased Jess and Keith and say Joey did the experiment right that we should have done all along. And we put these cages out all over the Mission Aransas Nurse. So we got out of Goose Island State Park and went all over. We did really rigorous measurements of turbidity with data recorders and Secchi discs and all kinds of stuff. And essentially, we wanted to really look at the role of turbidity and predation together. And this is Joey's data. So for those of you that aren't familiar with an MDS plot, each one of these points represents an entire sample, right? All the fish and crabs and shrimp. And the closer these points are together, the more similar those samples are. And the farther those points are away, the more dissimilar they are, okay? So two things to take away from this graph is the first one is the clear and the turbid sites are really different, right? Which wasn't really all of that surprising. But what was surprising to us was that the predator exclusion treatments, which are represented by these solid squares versus the open ones, which are the controls, the predator exclusion treatments have a big effect when the water is turbid and not so much when it's clear. And that may seem a little counterintuitive, but we think that that's because the predation in the clear sites is so high, there just aren't enough little extra crabs and things to find their way into our bioboxes because the fish eat them all long before they get to our treatments on those oyster reefs. So again, clear evidence that fish and turbidity are really important. The fish aren't the top predator, right? What's the top predator? We are, right? And so we catch these fish, right? Which then changes top-down control, which has the potential to change these food webs. So what we really needed to do was a people exclusion experiment. Well, Hurricane Harvey provided such an opportunity for us in 2007. So Harvey was a Category 4 storm, and it came in and it struck our field site. And when I say it struck our field site, I mean it struck our field site. We took a direct hit from the storm. Now, when that happened, it essentially removed fishing pressure and set up kind of a de facto marine protected area for about 12 months. So fishing pressure went way down. Rockport's a tourist destination. People weren't traveling there. People that lived there were rebuilding homes. They weren't doing a lot of fishing. Boats were destroyed, etc. Fishing pressure goes way down. At this time, I met Evan Pettis. He's become a good friend and collaborator who works for Texas Parks and Wildlife. What they do to set their fisheries catch and slot limits is they do these fisheries independent data. So they set gill nets to collect sport fish. They do these bay trawls to look at the food webs that the sport fish eat to judge the overall health of the bay. And they judge fishing activity, both by counting boats at boat launches and how many fishermen they survey on the water and so forth. And working with Evan, we noticed this pattern. So for those of you that might not be familiar with this term, this is catch per unit effort. So it standardizes the number of fish caught by some metric that of activity that you try to use to catch them. In this case, for the fish, this is the number of hours gill nets are in the water. So this is like fish caught per hour. And then the anglers, which are represented on this axis, are the number of people they interviewed when they were out on the water. And what you'll notice is after the storm in 2018, we see a much lower number of fishermen, right? And by the way, they're just counting boats and talking to people. It's not even clear those people were fishing. They might have been out there repairing a dock or cleaning something up. And we see a significant increase in the abundance of the two most common sport fish. We couple that with their surveys of the food web, and all of these things that these sport fish eat, like spots and croakers and pinnated shrimp, are significantly less after the storm when the fish populations are really high. Okay? So, huge effect of a natural disaster on behavior. Well, Joey and I went back out. We did the predator exclusion experiments again at Goose Island. This is our turbid site, and we got this pattern. So the predator exclusion still had an effect, but the whole community shifted, again, based on these fish. So I moved to Dolphin Island, and I thought we were done with the oyster reefs in Texas. And then COVID hit. And so we wrote a grant to go back to Texas and do this again because we thought COVID might change fishing activity, right? People couldn't travel, it was lockdown. And what we actually found was it slowed down fishing activity for a few months, and then it sped it up a whole lot later in the year because it was one of the few things people could do when things reopened. So COVID didn't have the big effect we thought it did. But while we were there, putting out our predator exclusion cages and clear and turbid sites, Winter Storm Uri hit. And this had the exact opposite effect of the hurricane because it killed all the fish. Somewhere in the neighborhood of 4 million fish in Texas died in the storm. Here you see trout and red drum, actually the ice on the fish, frozen. So a lot of fish died. So now we went from an abundance of fish to a reduction in fish. So we worked with Evan. We looked at their data. Again, the red is catch per unit effort of sport fish, which is on this axis. The blue is catch per unit effort of all those intermediate things that sport fish eat, pinnated shrimp and spots and croakers and bait fish. And what you'll notice is when sport fish abundance is high, those intermediate consumers are low. And then you see this change in that when the hurricane happens, right? It drives sport fish up and that goes down. And then here's the freeze, the fish die, their numbers go down, and those things go up. And we're still seeing a period of recovery now, 4 years after the storm. So two big natural disasters had really big effects on this food web. What's really interesting, though, is the oyster reefs responded to this disturbance differently, whether they were in clear water or turbid water, right? I know this is a lot to take in. So the solid shapes represent turbid sites. The empty ones represent clear sites. Colors are years. The squares are the exclusions, and the triangles are the controls. And so it's hard to kind of tell what's going on, except what you will notice is the clear and the turbid sites maintain their difference. Okay. So I took this data and I made what's called a Jacquard matrix from that, which gives me the center point of that group of that year, from where the average point is. When I do that, I get a pattern like this, right? So in the clear sites, you see how tightly coupled that is? Versus in the turbid sites, how these are spread out. So in clear sites, which have really high fish predation, which are more biodiverse, they're more resilient and resistant to the change than in the turbid sites where those factors are low. So not only do predators have a really big effect on oyster reef communities, they have big effects on resilience and resistance to disturbances, which is ultimately controlled by sensory ecology and how well the fish see and forage. So we know that reduced fish predation by exclusion or by population declines can cause significant changes in food webs. We know that turbidity affects predation rates, and that low biodiversity seems to be really good. Low turbidity promotes stability because it facilitates fish predation and maintains this higher level of biodiversity. We see that recreational fishing can have big effects on food webs, which makes it important to manage these things and for us to follow fishing regulations. And finally, we know that disturbances can significantly affect estuarine communities because it can modify top-down control. It can increase it or it can decrease it depending on how it affects not only the food web itself, but human behavior. All right, so we've covered that. Let's talk a minute about oyster reef chemical ecology, okay? This is a lot of the work that we've done here. So in this part of the talk, we want to talk about predators' effect on oyster morphology. So we noticed early on in work that Keith was doing and some work that Jessica was doing that oysters tended to grow heavier shells in areas where predation was high, especially when the water was turbid. So when there were lots of crabs, we got heavier oysters. So we wanted to know more about that, specifically how they responded to predator signals. We've been working to identify what it is the predators release that those oysters respond to, and we wanted to see if we could manipulate that somehow to improve restoration outcomes. So, through a whole bunch of different studies, the first of which was led by Elizabeth Robinson. Elizabeth is now with Louisiana Sea Grant. She'll actually be here giving a seminar in a few weeks. I encourage you to come and hear her present. But essentially, when we expose oysters to a caged predator, whether it's in the lab or in the field, we find that oysters grow a heavier shell that's also stronger. And we've done a ton of feeding experiments where we, in the lab and in the field, where we give predators a choice between oysters we've exposed to predators to make tougher and control oysters that we haven't. And the predators routinely eat more oysters that are not previously exposed. Okay, so it follows a trend like you would predict. In this particular graph, you can kind of see oyster responses to predators. So these are diploids, which are, I guess you'd call them kind of regular oysters, and then triploids are oysters that are bred for aquaculture. Okay, they have a third set of chromosomes. They're thought to be sterile, and so that they put more energy into growth. And we, in general, see that they grow faster. So you'll notice that the diploids are smaller than the triploids, but you'll also notice that the blue crab ones are smaller than the controls. So both diploid and triploid oysters respond, and they make a shell that's much, much harder, okay, in response to these predator cues. Now, responses to predators and oysters are not uncommon. We see them in a lot of different things. Probably the most well-known are tadpoles, right? So tadpoles normally are shaped like this, but depending on the type of predator around, they can grow a thicker tail or a bigger base to make them swim faster to get away from predators, or to have a larger structure that makes harder predators to eat them. And we see this in things like, here's a carp with a deeper body when pike predators are around. We see it in Daphnia, where they grow in spines. We see it in rotifers with spines, and with snails that grow thicker shells. So plasticity is really widespread, right, in a way for animals to deal with predation risk. And I've been a part of a couple of review papers where we reviewed somewhere in the neighborhood of 1,300 studies. And in over 90% of them, these plastic responses to predators were mediated by chemical cues. Okay, it's some signal the predator gives off that's chemically mediated that the prey respond to. And what we know is that those chemical cues are affected by several things. One of them is cue delivery, right? So think about, like, this is a chemical odor plume in a lab, in a laminar flow versus in a turbulent flow. And so you can see the turbulent is more mixed up, right? If turbulence mixes the chemical signal, it can make it harder for an organism to detect it and then ultimately to respond to it. We can see things like cue concentration, right? If there are more predators around, maybe the cue is more concentrated, so it's scarier. The frequency and the duration that organisms respond or are detecting these predators also has a big effect ultimately on how the degree to which they respond. And finally, and this is what I'm really going to talk about today, the type of cues prey detect have big effects on the way they make these decisions. Okay, because when you change your morphology in some way, you pay a cost to that, be it growth or reproduction or some combination thereof, to change something about yourself. So prey need reliable information about the location and intentions of predators to do that. So in all of our experiments, whether they be in the lab or the field, we have some predator or predator cue. It's like a caged crab. The crab releases something detected by the prey, in this case an oyster. The oyster then responds to that. Here's a cage in the field. Here's some mesocosm work we did at DISL. Here's some work we've done off docks in the field. So we've done this a lot of different ways, but the commonality is we're exposing oysters to some cue from a predator. We then bring those oysters back to the lab after a period of time, and we measure their shell area. We measure the weight of their shell, and then we use this device called a penetrometer where we crush the oyster and we record the maximum force it takes to break the shell as a measure of shell strength. And I'm going to focus really a lot on shell hardness for this talk because that's the thing that seems to be most relevant for them in surviving predation. So we wanted to ask several questions here. What are the costs associated with this? What's the nature? What actually is it that they smell? And then, of course, can we manipulate that for a practical outcome? So this is Emily Combs. Emily was my first REU student at DISL, and then Belgrade, who's given a talk like this up at Stony Brook today, who got this ball really started here. And so this is that diploid triploid picture I showed you a while ago, but look at the soft tissue, right? So organisms that are responding to predators are making harder shells, but they're growing less soft tissue because of that, which makes sense, right? You take in a finite amount of energy, you dedicate more to growing a shell, there's not enough left to grow soft tissue. So if there's a high cost to be paid by the oyster, is it really worth it to do this for restoration, right? Like if it makes them not reproduce as much, this could be bad. So we wanted to know what the long-term effects were of doing that. So we grew oysters here at the Sea Lab for a month, both diploid and triploid, exposed some to predators or not, and we worked with some private oyster farmers. We gave them these oysters and said, please grow these in your farm for us. Use whatever aquaculture techniques you use, whatever kind of cages you use, tumble them, clean them, whatever you do. Just grow them to reproductive age, which takes about a year, market size about a year. And we'll come out and measure them periodically, and we can see what long-term effects this has on growth. Well, we worked with French Herman Oyster Company in Mississippi, and then Point of Pines and Navy Cove here in Alabama. So you get a good spread across the Northern Gulf of various abiotic conditions and different farming techniques and so forth. But across all of them, what we really found was that once we got the oysters out of the Sea Lab and put into the farm, they grew out of it, right? So the ones that exposed to crabs, they recovered, they started to grow more like the controls, and they ended up with similar levels of soft tissue as those that had initially been exposed to predators. So we think that even though there's an initial cost to be paid, we think it's worthwhile because it helps get oysters through that initial period where they're small and where they're super, super vulnerable. But the question I always get asked is, well, what if you exposed them to predators for like the whole time, right? What does that look like? So Avery, my first student, one of my first students, Avery and I set this up in Texas, and it got knocked out by Tropical Storm Aaron. And then Ben and I set it up here, and it got taken out by Hurricane Sally. And then we tried one more time, and a bunch of them died. But finally, Jessica Helviker to the rescue. So I'm not going to go too much into this because she's going to present her prospectus talk in a few weeks. But Jessica has just completed a long-term induction experiment out on the West End. So we've been able to do this in the field for a year. And what Jessica did was she took oysters in the mesocosms here at the lab, and she grew them for a month, either with or without predators. And then she transferred them out to the West End. Some of those, never were exposed to predators at all. They were exposed to controls here in the lab and then hung in the field. Some of them were exposed to predators in the mesocosms here, and then she hung caged crabs beside them, so they kept getting that predator cue in the field. And then she had a third treatment where these were the ones, kind of like we did with those farms, where we gave them the initial predator cue in the lab and then moved them out to the West End and put them in an area without crab. And her data looks really interesting. So when it comes to soft tissue growth, right, what we see is that the ones that are, we'll call induced, that are exposed to crabs, have less soft tissue mass than the other two. The induced ones for a month are a little less than controls, but overall, the ones that were exposed to blue crabs are continuing to grow less over time. And likewise, those continue exposed to blue crabs are continuing to make a harder shell, unlike the twos that are grown in control. So yes, they do grow out of it, but if they're exposed to predators all the time, they will continue to develop a shell, which will slow their growth. So if you have an oyster lease, you want to keep predators off of it as best you can, right? Because they will have these long-term effects. All right. Well, what is it that these oysters react to, right? We know it's a chemical cue. What is that cue? What does it look like? What is it about? What are the characteristics of it? So this is Avery Shear. And Avery was a graduate student. She graduated in 2016 from my lab. And so she compared different kinds of cues that oysters might use to detect and evaluate predation risk. And we'll divide these into what we'll call direct cues, which are those released from the predator, versus indirect cues. And indirect cues are things that are released by injured prey, right? And so you would think that, like, if you can smell a predator, right, like, you know it's there. If you smell an injured prey, it might be because of a predator, but it might be because of something else, right? So we would think the direct cue would be somewhat more reliable than an indirect cue. So we expected that if we used like a blue crab predator versus like an injured oyster cue, that oysters might respond to both, but that they would respond to this one more than this one. But also on oyster reefs, we see a lot of clams that grow in the reef matrix, right? So if a crab is eating a clam, it could also turn around and eat an oyster. So maybe they'll respond to injured clams as well, right? But maybe not to the same level they would respond to an injured oyster. So the first experiments we did in this was we did sort of these indirect cues where we just basically crushed prey and injured them and exposed oysters to these injured prey cues. And then we did some direct cue treatments by feeding blue crabs either oysters or clams or starving them. And of course, we did controls. And as you would maybe expect, right? If we think of this as shell strength, right, by that crushing force metric we talked about, so the control oysters make a weaker shell than those exposed to these indirect injured prey cues, and those are not as strong then as those that are exposed to these direct cues, but both of these are stronger than the controls. So they respond to both. Okay, they do respond to both, and they make a stronger shell. Where it got weird though was when we broke it down and looked at them by treatments, right? So we saw that again, crushed clams and crushed oysters made the stronger shell, and blue crabs, of course, made an even stronger shell. But check this out: when we fed crabs clams, we got a stronger response than when we fed them oysters. And as with most things that you get in ecology, it feels like sometimes things just don't make sense, right? And we struggled with this one for a while. And the neighbor said, well, we buy the clams from a supplier and we keep them in the lab alive. And I bust open one of the valves and I feed it to the crab while they're alive. But to save space, I shuck the oysters in the field and I put them in a bag and we either refrigerate them or freeze them, and then I use tissue. So we repeated the experiment with help of Miranda, who was an undergraduate in the lab at the time. And we fed the crabs either fresh oysters or we fed them tissue we had aged for 72 hours. And lo and behold, they responded a lot more strongly when we fed them a living fresh oyster than we fed them oyster carrion. So an oyster can distinguish between a predator and a scavenger. What I almost called this talk was, for a snot-filled rock, oysters are pretty smart, right? They really fine-tune these responses to maximize like the benefit but minimize the cost. So we know that they react to these cues, right? But what are the cues? What are? And the challenge is that once those cues get released into seawater, they're at super low, even nanomolar concentrations. There's all these background chemicals in the water. So it's really hard to do chemistry in water, but it's even harder to distinguish those tiny concentrations of these predator cues from background noise. So what we needed to do was to get the cue out of the predator somehow before it went into seawater. So we looked at two crabs, stone crabs, blue crabs, and then two fish, sheep's head and black drum, that are known oyster predators. And we thought that urine would be a good place to target to get the cues out of, right? And that's what we've been doing. So this is me getting urine out of a blue crab. So get urine out of crabs is pretty straightforward. You chill them down on ice, and then you put a hollow gauge needle into the nephropore and you suck it out, and you get 1 to 4 mils at a time. And we call this milking. And we milk the crab for a while, and then we get all this urine, and then we can do experiments with that. The fish proved much more challenging. They were hard for me to catch. If Ronnie Baker was here, he would tell you about going out fishing with me, and he and Dylan Keene catching 24 sheep's head, and I'm not catching a single one. Every single volunteer that helped me with this project caught a fish before I did, including my own wife. Okay, but we finally got with the fishing guide, and he helped us catch a bunch of fish. But the fish were tricky, right? So we brought them into the lab. We would use tricaine to anesthetize them. And then we were able to do surgery on them where we put a catheter in, and we were able to harvest urine. And this is Eliza Kroom. She was in RU 2 years ago. This fellow in the back here trying not to pass out is my son Joseph. I like to call this picture the day Joseph decided to be a history major and not a marine science major. This was the day he abandoned STEM for history. So I need to give a special shout out to Jenny Bloodgood. For those of you that don't remember Jenny or didn't meet her, she was a really great veterinarian that was here as part of the Mammal Stranding Network and worked with the Carmichael Lab. But Jenny helped us figure out how to do all this, right? And so we would insert a catheter into the fish when it was under anesthetic. We would put some medical tape on the catheter itself, and then we would suture it in place so that the fish wouldn't hopefully pull it out. And then we could come out every day and use a syringe on the end of the catheter and harvest some urine from. Okay. So now that we've got sort of this urine, it allows us to do experiments that we really couldn't do before. Like we can really control the concentration of the predator, right? Because we know how much Q that they're getting. But because the urine is really time consuming to get, we can't really do it in a system where the urine could be flushed out into the water. So when we do that, we use these 2-liter jars. We often work in the mesocosm to keep them in a water bath so they don't get too hot. So this is a closed system. We feed them every day. We change the water and add the Qs a couple times a week. And then, of course, we add the urine in different concentrations or from different predators. So the first summer of 2019, Ben and I wanted to test to be sure that it was actually urine that we were seeing. So again, this is a measure of that crushing force, which is the shell hardness. This is seawater, which is our control. This is a positive control, what we'll call predator water. So we have a tank that has like a dozen blue crabs in it that we've been feeding oysters to all summer. So we know that it's like really scary and they'll react to it. And so this is like the weakest response we should see. And this is like the strongest response we should see. And then this was two blocks of urine that we collected from blue crabs. And so you can see that the urine is not quite as strong, but it's definitely stronger than the control and not statistically different than the positive control. So we know that it's really the predator urine, right, that's driving this. And it turns out when we look at shell strength, so again, this is our seawater control, and this is that predator water positive control. Chlorohex is an antiseptic we use during surgery, and we wanted to be sure that wasn't biasing the experiment, so we put some of that in some of the controls to see. But look at the urine from all 4 of those different predators, right? Causes these oysters to grow a significantly harder shell than we saw in controls. So we know that it's in the urine, and that gives us something to work with. So we're using a process called metabolomics, where we collaborate with these colleagues at Georgia Tech. And essentially what we do is we get some kind of response strength from a bioassay. So in this case, this is maybe shell hardening, and like this purple one is made a much harder shell than this red one is. We give those urine samples to Georgia Tech, and they run chemical analyses on them, and they get a series of these like peaks in an NMR. And then we can look at which peaks are present, as well as how big those peaks are when the response is strong versus when it's weak. And then that tells us what molecules are likely to be involved in that response. And then we can manufacture or buy those molecules and test them individually to figure out which ones cause the induction and which don't, and then what concentrations and combination. So this is a example of a readout, and this is a chemical signal here of something called trigonylene. Trigonylene is really common amongst all 4 of these predators. It's involved with amino acid protein breakdown, and so it's present in the urine of both vertebrates and invertebrates. And so the first 2 molecules we identified were trigonylene and something called homarine. Homarine is only present in invertebrates, so we found it in the crab urine, but it's really common. It's named for lobsters, which are in genus Homarus. But anyway, so we tested these 2 molecules, and so this was one of the things Elle did her first summer here. Was comparing like oysters grown in controls with crab urine to a mixture of these molecules. And what you'll see is when you get shell hardening of these, see how that response is kind of intermediate? So they kind of respond to it, but not really. And what that tells us is there's something else going on. And so we're still working with this. We found another molecule last summer, and when we add it with these 2, we get a stronger response. And so we're pretty close to nailing down, at least for blue crabs, what the signal is, so we can synthesize it, right? And then we can give it to, say, an oyster farmer to harden their oysters. So what cues are they using to evaluate risk? Well, these are products commonly found in urine. They tend to be things that are involved with some sort of metabolite. They're primary metabolites. They're involved in metabolism and breakdown, primarily protein. All right. Can we use it to improve aquaculture? Well, to do that, we wanted to actually shift gears just slightly because we've done a lot of work with blue crab induction on oysters. Um, But oyster drills are the main predator here, right? And crabs crush, and oyster drills actually kind of burrow in between the valves and inject a venom, and it makes the oysters relax and they eat them. But it's a very different mechanism. And so we wanted to know if the induction we got from crabs was similar to the induction we got from oyster drills, and if it worked similarly. So we figured out a couple of things as we were doing this. One was that we should settle oysters on these tiles because we could fit more of them into the mesocosm, and it allowed us to standardize the settlement area, and it was easier to measure and crush the oysters. So we worked with Auburn, and we settled oysters onto these tiles, and then we put them into mesocosms with either caged crabs or caged oyster drills. We grew them for a month, and then we put them out in the field. So up at Point of Pines area where the oyster farms are, that's like there's more oyster drills there than about anywhere I've ever been. Over in Portersville Bay, there are oyster drills here too, but it's a little fresher there, and so there's a lot more blue crabs. And then down on Dolphin Island, we have plenty of both. So we took those tiles, we attached them to rebar, and we sort of alternated which ones were at the top and which ones were at the bottom, and we put out like, God, 600 or 700 of these up and down oyster farms and all over the different sites. We used these predator exclusion cages to show that mortality was due to predators. And essentially what we found is all the sites, we got a significant increase in survival of oysters when we exposed them to both blue crabs and oysters. So we really didn't see a difference. This is data from just a week. Now, so you can already see like how much predation happens down here on Dolphin Island because the predation is really, really high. We left these out in the oyster farms for a year. And the ones that we left out, like I think the survival out of, I can't remember how many 10,000 we put out was less than 100, but all of the ones that were alive after a year were exposed to predators first, right? So it definitely works, and it can work long term. Hey, Krista, I found a good picture. So Krista has been working to do a big scale restoration project to see if we can actually use this to build an oyster reef. We've done this in Mississippi. We've done this a couple of times in Alabama. But essentially, we bag up literally tons of oyster shells. We put it in the mesocosms here. We try really hard not to get it into the drain lines because Matt gets really angry when we do that. We expose them to caged crabs or not for a month, and then we put them out. So this has been a couple of Eagle Scout projects and things that we've built these reefs with. This was the first one we did that Crystal had. And so this was done at Lightning Point and by La Battery. So we built reefs, and then we built them in columns. And so the red were ones grown in controls. The blues were grown with blue crabs, so they're stronger. We used these predator exclusion cages because we wanted to show that mortality we were seeing on this reef was caused by predators mainly. And largely it was. We did have some death and their predators could get in, and there was some crowding issues. But overall, the oysters in the cages grew well, and there was plenty of them. Mortality outside of the cages was really, really high. This is Krista's data that she just published. Like this paper literally just came out this week. And so essentially this is time, and then this is the number of spat per shell. But here's the number we really care about. So over a year later, we're seeing a 68% higher survival of oyster spat that we've put out on this reef, increased survival. So it's a good technique to do. It's pretty easy to add caged blue crabs when you're growing oysters for restoration. It toughens them up. It increases their survival. So it's a technique that we encourage for restoration activities. And in fact, Mississippi Department of Marine Resources is now doing this in their restoration program. All right, so I would say yes, shell plasticity can be used to improve restoration. It's a good technique, and we want to keep going. And we're hoping to figure out what those cues are. So then instead of using cage predators, we can just make like an IV bag of scary juice that you can dribble into your oyster tank to improve outcomes. All right, there's a ton of people to thank that have been involved with this. I can't possibly do all this work alone, and I'm grateful for all of them. And I'll end it here and take any questions. Thank you guys so much for coming.